Hey everyone, welcome back to Window Seat Studies. I know it's been long and we were discussing about immunology. So we started discussing immunology with an introduction about the immune system, the innate and adaptive immunity. Then we learned about the cells of immune system, the organs of immune system, and recently we started discussing deeply about the innate immunity. So we started discussing about pattern recognition receptors. So the previous lecture was a kind of introductory lecture about pattern recognition receptors. We learned what are pattern recognition receptors and how this idea was given by the researchers at that point of time. And in today's lecture, we are going to discuss about the types of the pattern recognition receptors, and we will be discussing about one particular type, the toll-like receptor, in detail, right? So, what are pattern recognition receptor? As discussed previously, these are the receptors or the sensors which are germline encoded, and they detect the pathogen associated molecular patterns and damage associated molecular patterns. So I discussed about these PAMs and DAMs in brief in previous lecture, but let's discuss about them in detail in this lecture. So basically, like if you have any idea about cellular signaling, then the pattern recognition receptor part will be quite easy for you to understand. So pattern recognition receptors are nothing but sensors or the receptors. So the job of receptor is to bind to a ligand and start some signaling pathway, right? So pattern recognition receptors, they are the sensors, means they are the receptors. So they are going to interact with some ligand, right? So the ligands or let's call the features or the patterns which they recognize, because they do not recognize a simple molecule, right? They recognize pattern, a particular pattern in the microbe, and that is why they are referred as pattern recognition receptors. So those pattern recognition receptors, they recognize some patterns which we refer as PAMs or DAMs. Okay, so you can have an analogy of like any receptor, so let's say like GPCR. So GPCR is a receptor which is going to bind to its ligand and it is going to start its signaling activity, right? Once the G protein gets activated. So in the same way, pattern recognition receptor needs to be interact, like the pattern recognition receptors, they interact with PAMs, the pathogen associated molecular patterns, or DAMs, and then they start their signaling activity. So pattern recognition receptors, they are part of innate immunity. So in last class, we learned that these pattern recognition receptors are the ones which are present in the Drosophila and they mount a sophisticated attack against the pathogen, right? Because Drosophila lack the adaptive immunity. So in the Drosophila, a toll protein was identified, and in the humans, there are certain proteins or the receptors which are similar to those toll proteins. So we refer them as toll-like receptors, which are again a part of pattern recognition receptor. So these receptors, they are going to recognize those PAMs and DAMs. So what are those? Okay, so they recognize PAMs, pathogen associated molecular patterns. So it is quite evident from the name that these are the patterns, okay, specifically the molecular patterns which are pathogen associated. Okay, this means these are the microbial features which are absent in the host cells. Let's take for example the lipopolysaccharides. So lipopolysaccharides, they are found in gram-negative bacteria, right? So they are only found in gram-negative bacteria. They are not found in the host. They are not found in our cells. That is why this is recognized as a foreign entity by those receptors, the pattern recognition receptors. So that is why we refer them as the pathogen associated molecular patterns. Then there are the double stranded RNA, which are the genetic material of the RNA viruses. Then there's peptidoglycan of the gram-positive bacteria, right? So LPS, double-stranded RNA, peptidoglycan of gram-positive bacteria, as well as the unmethylated CpG dinucleotides. These are again not not associated with the host. So these all of them, these are the features which are associated to different microbes, which will be recognized by pattern recognition receptors. So these are pathogen associated molecular patterns. Means these are the features which are being associated with the microbes. These are not found in the host cells. So they will be recognized by pattern recognition receptors. But besides this, the damage associated molecular patterns can also be recognized. So these damage associated molecular patterns, unlike PAMPs, means DAMPs, unlike PAMPs, these are the endogenous, means these are the found in host cells. So these are endogenous molecules which are released by the damaged or dying cells, right? So for an analogy, you can take, you can imagine certain government institute, or let's say that there's some lab, okay? So laboratories where where very important research is going on, or let's say it's some institute which is protecting some valuable information or valuable entity. So in such institutions or in such laboratories or in such settings, there is high security, right? So whenever this security is breached, whenever there's invader, the attack of invader, the alarm will go on. Like alarm will alert the whole institute, the whole security personnel and guards which are appointed there for security, that some invasion is there, right? So let's say that, imagine you might have seen this in movies, that some invader comes in such institute, right? So like they don't have the proper face ID or the fingerprint. So if the face ID which is associated with this invader, it is recognized as, okay, so this is not the usual face ID of the, like, of the people which are the part of institute. So this is some foreign face ID. So the alarm will go on, right? So this face ID or those fingerprints, they are associated with that particular individual, which is invader. So this invader is being recognized by the features which that particular person has, which that particular invader has. So we can say that this face ID or this fingerprint is pathogen associated molecular pattern, means it is associated to that invader, right? So let's say now the invader has invaded, okay, somehow. So like they always show some character which invades that institute. So the invasion has already been occurred, okay, and now when that particular person has invaded, let's say that he's killing the guards, or he's no, he's harming the guards. So in that situation, you might have seen that they inform the other guards, other security personnel with a walkie-talkie, or with the help of just, like, putting the alarm on. So they will be alerting the other guards, other security personnel, that invasion has been done. So there's some invader in our institute who's trying to steal the information, or who's trying to do the damage, right? So such type. So a guard informing other guard, it's damage associated molecular pattern. So in the same way, in our body, if there's invasion and the cells start dying, so those dying cells or those damaged tissue, so those damaged cells or dying cells, they will secrete some molecules. So these are the molecules which are secreted by the host cell to alert the other cells that there's invasion, that there's infection. So such type of molecules which are secreted endogenously, which are secreted by our body, these are known as danger associated molecular patterns, right? So I hope the difference is quite clear. What are PAMPs and what are DAMPs? PAMPs are the pathogen associated, whereas DAMPs are the endogenous molecules, right? So PAMPs will never be seen in the host cells, whereas DAMPs will be only seen in the host cell which has, which are damaged or which are dying. So for the example is ATP, okay, so ATP will be secreted, and then there are uric acid signal, even the mitochondrial DNA spilling out, all of those things, these are the damage associated molecular patterns. And then there is a protein known as HMGB1, which is also damage associated molecular pattern. If this protein is identified by the cells, this means that there is invasion. So the PRRs, the pattern recognition receptors, will be recognizing those PAMPs and DAMPs. So HMGB1, which is a DAMP, which it will be recognized by the pattern recognition receptor. So I hope the basic idea of what are pattern recognition receptors and what do they recognize is clear to you all. Okay, so yeah, the important point is that those PRRs, these are germ line coded. Okay. So unlike the receptors of adaptive immunity, let's take the T cell receptors or the B cell receptors. These are not germline encoded. So adaptive immunity receptors, these are, there's some genetic rearrangement, there's some recombination which is forming those receptor. Where on the contrary, those innate immune receptors, the PRR, pattern recognition receptors, these are germline encoded ones. So this is a really important point, and this is a really nice characteristics that PRR has. So there are major types of PRR, right? I told you in the previous lecture that there are some different types of PRRs depending upon their location as well as depending upon which type of ligands they recognize. So let's just list down the all of the different types of all of the major types of PRRs, okay, so that we will be learning about them one by one. Okay, so major types of PRR is the very first one which I discussed previously is TLR, the toll-like receptor. Okay, so the toll protein, they were first identified in Drosophila and they were, recognized as the pattern recognition receptors. So then in the humans, we found some proteins which are like the toll proteins, and that's why they were named as toll-like receptors. So we have TLR1, 2, 3 to TLR10. So in humans, there are different TLR proteins and they are located over the plasma membrane as well as they can be located on the endosomal membranes. Okay. So this TLRs, they recognize some ligands, they recognize the bacterial lipids, they recognize bacterial flagellin, and they can also recognize the nucleic acids, specifically the viral RNAs. Okay, so they can recognize viral RNA, DNA. So these are few ligands which will be recognized by TLRs. So depending upon the location, as well as depending upon the ligands they recognize, there are different types of pattern recognition receptors, right? So these receptors, they are always situated. They can be situated on the surface, like on the plasma membrane or intracellularly. Here you see TLR is present on the plasma membrane as well, and few TLRs, they can be present on the endosomal membrane. Then the other type of pattern recognition receptor is the CLR. Okay, CLR is C-type lectin receptors. Lectins, these are the ones which they bind to the polysaccharides. So C-type lectin receptors are the CLRs. We will not be discussing about CLRs in detail in this lecture. Well, they are also present over the plasma membrane. And these are lectins, right? So these are lectin receptors, means they are going to identify the carbohydrates which are present on the fungi or which are present on the bacteria. Okay, so CLRs, they are specific for the carbohydrates present on fungi and bacteria. For example, the tectin 1, tectin 2, these are the different types of CLRs. Then the third type of PRR is the nod-like receptors, the NLRs. NLR are nod-like receptors. Okay, so these nod-like receptors, these are present intracellularly. So these are present in the cytoplasm of the cell. Okay, so these are not present on the membrane, and they identify the bacterial peptidoglycan and the toxins. Okay, so NOD1, NOD2, NLRP3, all those are different types of NLRs. They recognize the bacterial peptidoglycan and toxins. Okay, then there is different type of PRR, the other type known as RLR or Rig I-like receptor. Okay, so those Rigi-like receptors, they are also present intracellularly. So they are cytosolic ones. Okay, and they recognize the nucleic acids. So specifically they recognize the viral RNA. So they are really important to mount the immune responses against the against the virus. So these are antiviral ones. Okay, so then there's other type as well besides those ones. So there's other type of the PRRs which are referred as ALRs or AIM2 like receptors. Okay, so these receptors, these are also present in nucleus as well as cytoplasm. Okay, so these are really specific. These ones are present in nucleus, or this can be present in the cytoplasm. And again, this also those ones, they also recognize the nucleic acid, so they recognize the double-stranded DNA, specifically the cytosolic double-stranded DNA. Okay, so these are the different types of, like, the major types of pattern recognition receptors that we will be discussing. So we will be discussing about toll-like receptors in detail, nod-like receptors in detail, as well as the regal-like receptors in great details. In today's lecture, we will be only focusing on toll-like receptors. We will discuss about NLR and RLR in some other lecture. Okay, so let's learn about the TLRs. What are they and what do they do? So TLRs are toll-like receptors. So these are the receptors which resemble to the toll proteins found in the Drosophila, right? So those toll-like receptors, they are of different types. So there's not like the different types of TLRs are found in humans, like TLR 1, 2, 3, up to 10. Okay, so there are different types of TLRs in humans based on what type of ligand they are recognizing. So they can recognize they can recognize bacterial flagellin, or they can recognize even the viral RNA or a viral DNA. So depending on that, they have been categorized into different types, the TLR 1, 2, 3, 4. Okay, and they are again present in on the plasma membrane, as well as they can be present on the endosomal membranes. But before learning about those different types, the particular types and the particular ligands that they interact with, let's first learn about the, like, TLR system. Like, how are those, like, what are the different domains of those receptors? So the toll-like receptor, let me just show you how do they look like. So let's assume that this is the membrane. Okay, this could be plasma membrane, or this could be endosomal membrane. So on this membrane, I'm drawing a very simple schematic. Okay, so on this membrane, this particular receptor is TLR, the toll-like receptor. So you can see three clear domains. Okay, so this is the first domain, which is rich in leucine, rich in leucine repeats. So this particular domain is rich in leucine rich repeats. So this has leucine rich repeats. Okay. So this leucine rich repeats will be referred as LRRs. So LRRs are quite abundant in in this region. So like 18 to 25 these repeats are present in this particular extracellular domain. 18 to 25 LRR are present, which almost has 20 to 25 amino acids. Okay, so these are leucine rich, and this is the extracellular domain, right? So the job they have is to interact with ligand. So this LRRs, leucine rich repeats, they are going to interact with ligands such as the viral RNA or the bacterial flagellins. Okay, whereas the second domain is the transmembrane domain. So the TLRs, they have single pass transmembrane domains. Okay, which is going to hold them on the membrane. Whereas here you can see the intracellular domain, right? So this intracellular domain is, it resembles to the receptors of IL-1. Okay, so these are the ones which are going to resemble to the IL-1 receptors. So receptors which interact with the interleukin 1. And that is why, and these are found, this intracellular domain resemble to this IL-1 receptor, and this is found in toll, and that is why this is referred as TIR domain. Okay, so TIR means toll IL-1 receptor domain. Okay, so this is toll IL-1 receptor domain. So the receptors for IL-1, the interleukin 1, they have this intracellular domain, and this particular domain found in toll proteins, toll receptors, this resemble to that particular domain a lot, and that is why it is referred as TIR domain. And TIR domain is really important because it is the one which is going to initiate the signal transduction. So just like any other receptor, if you have seen my cellular signaling lectures, just like any normal receptor, the TLR also has their structure. The extracellular domain is going to interact with the ligand. The transmembrane domain is going to hold the receptor over the membrane, and the intracellular domain is the one which has the signaling activity. So just assume by some method we just cut this intracellular domain and we cleave this intracellular domain, then the activity of TLR will be completely abrogated. Okay, so this is the different domains of TLR, and the TIR domain is the one which is found in all of the TLRs, and it is the one which is going to carry out the signaling. So there are different signaling pathways which are mediated by TLRs, such as there is MyD 88 dependent pathway, and then there is TRIF dependent pathway. But we will be discussing about those signaling pathways in detail in some other lecture. Today we are just discussing about toll-like receptors, their structure, and how, like, what are the different types of TLRs and what are the different ligands that those particular TLR recognize. Okay, that's only topic of discussion. We will discuss about those MyD 88 dependent pathway as well as TRIF dependent pathway, their step-by-step mechanism, how they detect LPS, and how they dimerize, and how they do the kinase activation to do some downstream signaling, and what are the exact biological roles in next lecture. Okay, so let's now discuss about the types of TLRs. Okay, the different types of TLRs which are found in humans, which are found in our system. So like, on the basis of location, PRRs are differentiated into different types. In the same way, TLR are also of different types. For example, TLR1, TLR2, TLR3, TLR4, then there's TLR5, TLR6, TLR7, TLR8, 9, and 10. Okay, so these are different TLRs. So these different TLRs, they are located on the different positions. For example, TLR 1 and 2, both of them, they are present over the cell surface. So like, they are present on the plasma membrane. Whereas TLR 3 is present over the endosomal membrane. Okay, but TLR 4 is again present on the cell surface. TLR 5, 6, even the not just TLR4, TLR4, 5, 6, all of them, they are present over the cell surface or the plasma membrane. Okay, whereas TLR 7, 8, 9, they are present on the endosomal membranes, and TLR 10, they are present on cell surface. Okay, so if you have to categorize the TLRs which are present in the plasma membrane, whereas TLRs which are present on the endosomes. So the TLRs which are present on cell surface are 1, 2, 4, 5, 6, whereas the ones which are present over the endosomes are 3, 7, 8, 9. Okay, and even 10 is present on the plasma membrane. So these are the different TLRs which are present over the different locations. And those different TLRs, again, they recognize different ligands. They recognize different microbial features. For example, the TLR1 and TLR2 dimers, they recognize the triacyl lipopeptides, which are again present on the bacteria. So TLR1 and 2, both of them, they recognize triacyl lipopeptides, which are present over the bacteria. Okay, so these are the bacterial features which will be recognized by the TLR1 and 2. Then TLR3 is very specific because it recognizes double-stranded RNA, which is the genetic material of RNA viruses, right? So this is how the innate immunity, like the innate immunity which has pattern recognition receptor, in that particularly TLR has the capacity to recognize bacteria as well as recognize viruses. So it recognizes bacteria, the triacyl lipopeptides, the TLR1 and 2 does that job, whereas TLR3 is very specific for the double-stranded RNA of viruses. The TLR4, it recognizes the lipopolysaccharides of the gram-negative bacteria, okay, whereas the TLR5, they recognize the bacterial flagellin. Okay, so here you can see how the different, how the different TLRs, they have different ligands for the interaction. So they are going to recognize the different patterns. They are going to recognize different molecules which are associated to either bacteria or to viruses, right? And that is why these are the different types of TLRs. And again, their locations are also different. So TLR6, it recognizes diacyl lipopeptides. Okay, so these are the bacterial, bacterial features, whereas TLR7 and 8, both of them, they also recognize the, just a second. So TLR7 and 8, both of them, they recognize ssRNA, the single stranded RNA, which is again specific to the viruses. Okay, so single stranded RNA, double stranded RNA, even the bacterial lipopeptides, the lipopolysaccharides, all of them, they will be recognized by the TLRs. Then TLR9, that will be present on the endosome, and it is going to recognize CpG dinucleotides which are unmethylated. Okay, so unmethylated CpG dinucleotides will be recognized by TLR9, and TLR10 has kind of immunomodulatory role, and it is still being studied. Okay, so these are the different, different ligands with which the different TLRs will be interacting. So a few TLRs are really important for you to learn, and those are the TLR3. They will be recognizing the double-stranded RNA viruses. The TLR7, 8 recognizing the single-stranded RNA viruses, and TLR9 which are recognizing unmethylated CPGs, the CPG dinucleotides. Okay, so like you need to remember this in such a way, like TLR, there are few present over the surfaces and a few are present on the endosomal membranes. So the endosomal membrane TLRs are really important for you to learn and to remember for like in the CSIR perspective. So you can remember it in a way like the plasma membrane TLRs, they are bacterial specific, okay, whereas the endosomal TLRs, specifically 3, 7, 8, 9, these are the ones which are present over the endosomal membrane. So 3, 7, 8, they are specific for viruses. Okay, so the 3 recognizes double-stranded RNA, whereas 7, 8 recognizes single-stranded RNA, and 9 recognizes CpG dinucleotides. So these 4 are really important for you to learn. So with this, I'm done with the TLRs. So we have discussed about TLR in really detail. We will be discussing about their signaling pathways, the MID88 signaling pathway and the TRIF dependent signaling pathway in detail in next lecture. Till then, see you. Bye-bye.